Good day, everyone. This session, I'm going to discuss the enzymes and vitamins. This lecture is taken from the General Organic and Biological Chemistry of Stephen Stocker. To the general characteristics of enzymes, enzymes are usually proteins that acts as biological catalysts. When we say biological catalyst, it is the one who speeds up the reaction in the living system. As to background, each cell in the human body contains thousands of different enzymes, and these enzymes cause cellular reactions to occur millions of times faster than corresponding uncatalyzed reactions. The question now is that how these enzymes speeds up the reaction. So this enzyme speeds up a reaction by lowering the activation energy, okay, and changing the reaction path that provides a lower energy route for conversion of substrate to product. So enzymes as catalysts are not consumed in the reaction, and there are few enzymes are now known to be ribonucleic acids or the RNA. As you can see in this illustrations, we have two reactions here. One reaction that colors black, and another reaction the color red or pink. So this reaction, the black line here, represents the reaction without the enzyme, and this one represents the reaction with enzyme. So as you can see, the reaction without enzyme, it has a higher activation energy. While this reaction, it activates faster because the activation energy lower, thus it can produce products faster compared to the reaction without the enzyme. Now, we have the enzyme structure. One enzyme structure is a simple and conjugated enzymes. So most enzymes are usually globular proteins, and some are simple proteins, and other are conjugated proteins. When we say simple enzyme, these are enzymes composed only of protein or the amino acid chains, and it is the third three-degree structure of the simple enzymes that makes it biologically active. We have also the conjugated enzyme. This conjugated enzyme has a non-protein part in addition to a protein part. So here we have the apoenzyme and the cofactor or the coenzyme. When we say apoenzyme, these are protein part which are inactive in itself, while we have the cofactor or the coenzyme. These are non-protein organic or inorganic moiety which the activator and usually loosely bound to proteins. That is why they are called as coenzyme or the co-substrate, and it's inorganic, that is cofactor. So apoenzyme plus cofactor, it will form a holoenzyme, which is a biologically active conjugated enzyme. So in this illustration, it shows the apoenzyme, which is the protein portion, which is said to be inactive. And it will be, if we add the coenzyme or what we call the cofactor, which is the non-protein portion, and it is said to be the activator. So if we add this coenzyme to our apoenzyme, which is inactivated, it will form a whole enzyme active, which is known as the holoenzyme, with the presence of this substrate. Okay, the same explanation here, but different illustration. As to its nomenclature and classification, so most commonly named with reference to their function, the enzyme type will be classified or named by its reaction catalyzed, and it will be identified of the substrate. So a substrate is the reactant in an enzyme catalyzed reaction. So the substrate is the substance upon which the enzyme acts. Okay? For example, in the fermentation process, the sugar is converted to alcohol, and therefore, in this reaction, sugar is the substrate. And we have three important aspects of the naming process of our enzymes. One is the suffix, second is the type of reaction catalyzed by the enzyme, and the third one is the addition to type of reaction. So first, we have the suffix. The suffix ase identifies it's an enzyme. For example, we have the urase, the sucrase, and the lipase are all enzyme digestions. Okay, I mean enzyme designations. The ase here, the ase here signifies that this is an enzyme. Exception for this is that the suffix in or the in is still found in the names of some digestive enzyme, like for example the trypsin, the chymotrypsin, and the pepsin. So still, the trypsin, the chymotrypsin, and the pepsin are digestive enzymes that ends with in. So that's why they are considered as exception in the names of the enzymes. That most of the enzymes ends with the the ase, like urase, sucrase, lipase. Second one is that the type of reaction catalyzed by an enzyme is often used as the prefix. Okay, so when we say prefix, that means before. Okay, before the name. So, like for example, we have the oxidase. The oxidase actually catalyzes an oxidation reaction, and we have also the hydrolase. These are enzymes that catalyze a hydrolysis reaction. Okay. The third one, we have the identity of substrate is often used in addition to the type of reaction. So example, the glucose oxidase, the pyruvate carboxylase, and the succinate dehydrogenase. So these are the different examples of an enzyme that that their identity of the substrate is often used in addition to type of reaction. Just like, for example, we have here, we may predict the function of the following enzymes. We have the maltase, lactate, hydrogenase, the fructose oxidase, and the malate isomerase. So we predict what will be the function of these following enzymes. For letter A, it's hydrolysis of maltose. Okay, it hydrolyzes the maltose. Your enzyme is maltase. In letter B, lactate dehydrogenase. It is the removal of hydrogen, dehydrogenase from lactate ion. So that means in a lactate ion, this lactate dehydrogenase acts on the removal of the hydrogen in lactate. So next, we have oxidation of fructose for the letter C, because fructose oxidase acts the oxidation of fructose. We have here from the prefix, the oxy, that means oxidation, and it oxidizes the fructose. Then letter D, we have the malate isomerase. This is responsible in the rearrangement, or what we call in chemistry isomerization of the malate ion. Okay, so these are different types of enzymes with their functions. So that means when we say these, when we see these enzymes, it tells us on their functions, and at the same time, it gives us a hint with their names. And we have different major classes of enzymes, and they are six. They are six. These enzymes are grouped into major classes based on the types of reactions they catalyze. Number one, we have the oxidoreductases. when we say oxidoreductases, this actually um, an oxidation reduction reaction. Next, we have the transferases. In transferases, the functional group transfer reaction. That's why gintawag siya nga transferase. Next, we have the hydroxylases. Your hydroxylases, it hydrolyzes the the substrate. Okay, usually it undergo hydrolysis reactions. We have the lyases. We have the reaction involving addition of a group to a double bond or removal of a groups to form double bonds. Next, we have the isomerase. This isomerase, um, the reaction is isomerization reactions. We have the ligate. That means reactions involving bond formation coupled with the adenosine triphosphate hydrolysis. So these are the six types or six major classes of enzymes based on the types of reactions they catalyze. In this table, it shows the different classes and subclasses of enzymes. Like for example, we have the oxidoreductases. The subclasses are oxidases, reductases, and dehydrogenases. When we say oxidases, the type reaction it catalyzes, it oxidize of the substrate. We have the reductases, reduction of a substrate, and we have the dehydrogenases. It is actually the introduction of double bond or the oxidation through oxidation by formal removal of the two hydrogen atoms from the substrate, and the hydrogen is being accepted by a coenzyme. We have here the transferases. We have different subclasses, the transaminases, we have the kinases, and the functions, the transfer of an amino acid group between substrate for the transaminases. We have also in the kinases, we have transfer of a phosphate group between substrates. Next type is, next class is the hydrolases. We have subclasses, lipases, that hydrolysis of ester linkages in the lipids. We have the proteases, hydrolysis of amide linkages in the proteins. Nucleases, it's hydrolysis of sugar phosphate ester bond in the nucleic acids. We have the carbohydrases, the hydrolysis of your glycosidic bonds in carbohydrates. And we have the phosphatases, the hydrolysis of phosphate ester bonds. Another main class is the lyases. The subclasses are um, the, the hydratases, which it removes water from a substrate. We have the decarboxylases. We have the removal of carbon dioxide from a substrate. We have the deaminases, the removal of the NH3, the amine group from a substrate, and the hydratases, the addition of water to a substrate. Next main class is the isomerases. Subclasses are the racenases, the conversion of the D isomer to L isomer or vice versa. And we have also the mutases, the transfer of a functional group from one position to another in the same molecule. Thus, it is said to be the isomerases because it transfers these structures to a different form of isomer. Lastly, we have main class, which is the ligases. These are the synthases and the carboxylases. When we say synthases, is responsible for the formation of a new bond between two substrates with the participation of the adenosine triphosphate. The carboxylases responsible for the formation of new bond between a substrate and the carbon dioxide, with participation of the adenosine triphosphates. This time, I'll be discussing to you the models of enzyme action. The explanations of how enzymes function as catalysts in biochemical systems are based on the concepts of an enzyme active site and enzyme substrate complex formation. When we say active site, the active site is relatively small part of an enzyme structure that is actually involved in catalysis. The active site here happens where substrate binds to the enzyme. So this is the active site. And it is formed due to folding and bending of the proteins, and usually a cursive-like location in the enzyme. And some enzymes have more than one active sites. So again, this is the active site of an enzyme that is relatively small part of an enzyme structure and that is actually involved in catalysis. Next is the enzyme substrate complex. And we have the term intermediate reaction species. The intermediate reaction species formed when the substrate binds with the active site. So this is the active site and this is the substrate. Once the substrate binds to the active site, it will form the term intermediate reaction species, and it is needed for the activity of the enzyme. And the orientation and proximity is favorable and reaction is fast. That means the proximity and the orientation of the substrate to its active site will be favorable and the reaction is fast to occur. Now we have two models for substrate binding to enzyme. One is the lock and key model and one is induced fit model. Let us discuss first the lock and key model. The lock and key model, wherein in this model, the reaction site in the enzyme has a fixed, rigid, and geometrical formation. Okay? And the substrate here, the substrate of a specific shape can bind with the active site. So this is the active site of the enzyme, and the substrate must be specific or must be the same with the active site or the structure of the or the shape of the active site. A substrate whose shape and chemical nature are complementary to those of the active site can interact with the enzyme. So this one, for example. So once the substrate is able to bind with the active site, and if the active site and the substrate is complementary, it will react and form the product. Fails to take into account proteins' conformational changes to accommodate a substrate molecule. So this is actually one example. The enzyme, the functional shape, and this is the substrate. Okay? So once it enters, it opens the the lock. So this is what happened. So the substrate here is complementary. I mean the substrate um, shape and the enzyme's active site is complementary, then it will undergo a reaction and it will form a product. On the other hand, we have the induced fit model. So in this model, the substrate contact with enzyme will change the shape of the active site. Okay? It allows a small change in space to accommodate substrate. And for example, it is how a hand fits into a glove. So that means the substrate will contact with the enzyme and later on will change the shape of the active site following the shape of the substrate. And the enzyme active site, although not exactly complementary in the shape to that of the substrate, is said to be flexible enough that it can adapt to the shape of the substrate. Thus, it will produce products. Unlike to what happened in the previous slide in the lock and key model, the lock and key model is more specific compared to the induced fit model. Because, again, when the substrate contact with the enzyme, it will change the shape of the active site, giving the product possible. And that is why it is said to be flexible enough that it can adapt to the shape of the substrate. And now, what are the forces that determine substrate binding? We have the hydrogen bond, the hydrophobic interaction, and the electrostatic interactions. So notice in this illustration, the color yellow here is or are the R group interaction that bind the substrate to the enzyme active site. So this is the active site actually of the enzyme, and this is your substrate. The color green here are the R group interactions that maintain the three-dimensional structure of the enzyme. That means it produce it maintain the structure of the enzyme in three-dimensional way. And the color blue here is the non-interacting R groups that help determine the solubility of the enzyme. And now let's go to the enzyme specificity. We have different enzyme specificities. One is the absolute specificity. When we say absolute specificity, the enzyme will catalyze a particular reaction for only one substrate. That's why it is absolute. This is most restrictive of all specificities and usually not common. For example, we have the catalase is an enzyme with absolute specificity for hydrogen peroxide or the H2O2. We have also the urease, absolute specificity for urea. Another, we have the stereochemical specificity. This stereochemical specificity is an enzyme can distinguish between the stereoisomers, and it focuses on its chirality. That is why chirality is inherent in an active site where the amino acids are chiral compounds. Like, for example, we have the L-amino acid oxidase that catalyzes reactions of L-amino acids but not on the D-amino acids. That is why it is stereochemical specificity. It is more specific on its structural form. Another specificity is a group specificity. When we say group specificity, it involves structural similar compounds that have the same functional groups. They have the same functional groups. Like your carboxypeptidase. Your carboxypeptidase, the cleaves amino acid one at a time from the carboxyl end of the peptide chain. Another, we have the linkage specificity. Linkage, it involves a particular type of bond irrespective of the structural features in the vicinity of the bond. Considered most general of enzyme specificity, example, we have the phosphatases. It hydrolyzes phosphate ester bonds in all types of phosphate esters. So what are the factors that affect enzyme activity? These are the main four factors that affects enzyme activity: the temperature, the pH, the substrate concentration, and the enzyme concentration. First, we have the temperature. In the temperature, higher temperature results in higher kinetic energy, which causes an increase in the number of reactants collision. So therefore, there is high activity. When there is increase in temperature, there is a high activity of enzymes. But there is what we call the optimum temperature. This optimum temperature is a temperature at which the rate of the enzyme catalyze reaction in maximum. Over that optimum temperature, it will go down the reaction of the enzyme. The optimum temperature for human enzymes is about 37 degrees Celsius of the body temperature. That is why we have the body temperature of 37 degrees Celsius. Increased temperature, like for example in the case of with a person with high fever, will lead to decrease the enzyme activity. Looking into this illustration, the reaction rate increases when the temperature increase, but it should be um, fall into its optimum temperature. But beyond its optimum temperature, it will decrease its activity. Why? Because remember that enzymes are proteins. In higher temperature, proteins denature. That is why denaturation due to excess heat will lower the enzyme activity. Okay? Next, we have the pH. Drastic changes in pH can result in denaturation of proteins. Again, we focus our attention that enzymes are proteins. That's why drastic changes in pH can result to denaturation of proteins. And there is another optimum pH that the enzyme activity increase. So the pH at which enzymes has maximum activity is the term, it describes the optimum pH. Most enzymes have optimal activity in the pH range of 7 to 7.5, except for the digestive enzymes like the pepsin and trypsin in our stomach. It has the optimum pH of 2, and for the trypsin, I mean for that is for pepsin, but for trypsin, the optimum pH is 8.0. So that means the once the pH reach into its optimum, there will be a maximum activity of enzyme. And usually the optimal activity in the pH range 7 to 7.5 for the enzyme to be activated. Except for these different digestive enzymes present in our stomach, the pepsin and the trypsin. Okay? Let's look into this illustration. The reaction rate increases when the pH increases, but it should it should be on the limit of its optimum pH. Beyond the optimum pH, it will decrease again the enzyme activity because it will denature the protein. Okay, so this is the illustration that the provides us on understanding how the pH really work in the activity of the enzyme. Another factor is the substrate concentration. At a constant enzyme concentration, the enzyme activity increases with increased substrate concentration. Okay? In a constant enzyme concentration, the enzyme activity will increase if the substrate concentration increases. And there is an enzyme saturation what we need to understand. The enzyme saturation here, it is the concentration at which it reaches its maximum rate and all of the active sites are full. So that means if the active sites are full, we could say that the enzyme is saturated already. Okay, no more substrate can can attach to the active sites because the available active sites are full already. That means they have already the partner, but that is why it ends up with the enzyme saturation. And we have the term turnover number. So the turnover number, it is the number of substrate, the molecules converted to product per second per enzyme, and the molecule under condition of the optimum temperature and the pH. So in this illustration here, the reaction rate or the velocity of the reaction rate increases, the substrate concentration increases, the activity here of the reaction or of the enzyme activity increases until it reaches its maximum. That means it reaches the enzyme saturation wherein all of the active sites are full already. So that will stop the activity of the enzyme. Another factor is the enzyme concentration. The enzymes are not consumed in the reaction, they catalyze, and you should remember that. At a constant substrate concentration, the enzyme activity increases with the increase in enzyme concentration. Okay? So the greater the enzyme concentration, the greater the reaction rate. So that means lesser enzyme concentration, lesser enzyme activity. Eh, Higher or greater enzyme concentration, the greater the reaction rate. So this is the summary of um, the lessons that I've discussed earlier. The mechanism of enzyme action, the lock and key model, induced fit model. Okay? So when we say lock and key, it's specific. The induced fit model, usually it's flexible shape that can change its structure. Okay? And we have the factors that affect the rate of enzyme activity, the temperature, the pH, the concentration of substrate, and the concentration of enzyme. We have another enzyme known as the extremozymes. And this is actually produced by extremophiles. Um, Extremophiles organisms that are organisms that thrive in extreme environments. When we say extreme environments, these are hydrothermophiles that thrive at 80 degrees to 122 degrees Celsius in high pressure. So we have the acidophiles, the alkaliphiles, the halophiles, the piezo and the cryophiles. Usually, these are organisms lives in a very in extreme environments, and these are usually microorganisms. Okay, microorganisms like bacteria, like fungi, present in the thermal vents or maybe in the seafloor. So these organisms produce extremozymes. So extremozymes, which is a microbial enzyme that is active at condition that would inactivate human enzymes as well as enzymes present in most other organisms. Extremozymes are high interest for industrial chemists because enzymes are heavily used in industrial processes, and industrial processes require extremes of temperatures, pressure, and pH. And we have several applications of extremozyme. So biotechnology industry, production of enzymes for industrial application. We need this also for petroleum industry, the oil well drilling operations. We have environmental scavenging and removal of heavy metals, for bioremediation, environmental cleanup using genetically engineered extremophiles, and laundry detergents used in cold wash cycles. So these are very important enzymes that applicable in our day-to-day life, especially when we improve our industry. Now let's talk about the enzyme inhibition. The enzyme inhibitions are actually the inhibition of the activity of the enzyme. and we have the enzyme inhibitors. Enzyme inhibitors are substances that bind to an enzyme that actually stop or slows down the normal catalytic activity of the enzyme. So that's why we call it inhibitors, enzyme inhibitors. And we have three types of enzyme inhibitors: the competitive enzyme inhibitor, we have the non-competitive enzyme inhibitor, and the irreversible enzyme inhibitor. When we say competitive enzyme inhibitor, the molecule closely resembles the substrate. Okay, this is a substrate here, and we have the competitive inhibitor. They are the same in shape with the substrate. That's why it competes with the substrate. Okay, binding of this competitive inhibitor to the active site of the enzyme will temporarily prevent the substrate from occupying it, thus blocking the reaction of or the enzymatic activity. Or let me say that it blocks the reaction of the enzyme, because this competitive inhibitor that is resembling the substrate will stop or slow down the normal activity of the enzyme because it competes with the substrate. So that is the competitive enzyme inhibitor. We have the number two, the non-competitive inhibitor. So this non-competitive inhibitor or enzyme inhibitor, a molecule that binds to a site on an enzyme that is not the active site. That is why it is said to be non-competitive, because instead it interact with the with the binding site or the active site of the enzyme. It binds to the other side. It occupies the other side of the enzyme. Okay? So the normal substrate still occupies the active site here, but the enzyme cannot catalyze the reaction due to the presence of this inhibitor. Okay? Supposed to be this inhibitor should interact here, but since the shape is different, it will interact on the other side of the enzyme. But what will happen? It still inhibits the reaction of the enzyme. Even the substrate fits with the active site, because the reaction due to presence of inhibitor limits the activity. That's why it should not be here. That is why it is said to be an inhibitor, even though it is not competitive with the substrate. Still, it has an effect on the enzyme activity. The third one, we have the irreversible enzyme inhibitor. The irreversible enzyme inhibitor, it's a molecule that forms a covalent bond to a part of the active site, permanently preventing substrates from occupying it. Okay? It is not competitive inhibitor, but it is said to be irreversible inhibitor because this molecule forms a covalent bond on the active site, on the part of the active site. That is why it permanently prevents the substrate from occupying it. That is why the substrate cannot bind on this area, and it will inhibit the activity of the enzyme. Again, we have three types of enzyme inhibitors: competitive enzyme inhibitor, we have the non-competitive, and the irreversible enzyme inhibitor. Now let's proceed with the general characteristics of vitamins. When we say vitamin, it is an organic compound which is essential for proper functioning of the body and must be obtained from our dietary sources because human cannot synthesize them in enough amount. That is why vitamins can be obtained from the dietary sources. That's from our food that we intake. And vitamins needed in micro and milligram quantities. 1 gram of vitamin B, for example, is sufficient for 500 people. Okay, that's why we need it in micro and in milligram count quantities only. Enough vitamin can be obtained from a balanced diet, meaning we eat a balanced food. Okay. Supplemental vitamins may be needed after illness. Okay, that is what, because we haven't eaten a plenty of food. That is why we need to supplement vitamins. Many enzymes contain vitamins as part of their structures that are conjugated, that are said to be a conjugated enzymes. We have two classes of vitamins, the water soluble and the fat soluble vitamins. That is why when we say water soluble vitamins, oh, we take these vitamins with water. Okay, we take fat soluble vitamins with food. Okay, synthetic and natural vitamins have the same function, and we have the 13 known vitamins. Okay, so we have the two classifications of vitamins, the water soluble vitamins and the fat soluble vitamins. Vitamin C for water soluble vitamins, vitamin C, thiamine, riboflavin, niacin, pantothenic acid, vitamin B6, biotin, folate, and vitamin B12. Fat soluble vitamins are vitamin A, D, K, and E. Okay, so these are the different vitamins that are classified into water soluble vitamins and fat soluble vitamins. The table here shows the general properties of the water soluble vitamins and fat soluble vitamins as to its absorption, transport, storage, excretion, toxicity, dosage, frequency, and relationship to coenzymes. For water soluble vitamins, that includes vitamin B complex and the vitamin C. And for for fat soluble vitamins, the A, D, E, and K. For the absorption, water soluble vitamins are directly into the blood, while fat soluble vitamins will first enter into the lymph system. As to transport, it travels without carriers, while the fat-soluble vitamins, many requires protein carriers. And as to storage, water-soluble vitamins circulate in water-filled parts of the body, while the fat-soluble, from its word, fat-soluble, found in the cells associated with fat. As to its excretion, the kidney remove excess in the urine. That is why when you take vitamin C, excess of vitamin C will be expelled in our body through the kidney and excreted in urine. While the fat soluble vitamins tend to remain in the fat storage sites. How about the toxicity? The toxicity is not likely to reach toxic levels when consumed from supplements, the water soluble vitamins. While in fat soluble vitamins, likely to reach toxic levels when consumed from supplements. That is why it should be taken with prescription, especially the fat soluble vitamins. As to dosage frequency, needed in frequent doses are the water soluble vitamins. And for the fat soluble vitamins, needed in periodic doses. As the relationship to its coenzyme, it is function as coenzymes, while fat soluble do not function as coenzymes. So these are the general characteristics of the water soluble vitamins and fat soluble vitamins. Here we have the vitamin C as representative of water soluble vitamins. Humans, monkeys, apes, and guinea pigs need dietary vitamins, especially vitamin C. And the vitamin C, if we are deficient with vitamin C, um, it will it will produce a vitamin C deficiency known as scurvy. The scurvy is a disease whose symptoms include skin lesion, fragile blood vessels, loose teeth, and bleeding gums. It involved in metabolism of a certain amino acid. Another, we have the vitamin B or the B complex. The B here the thiamine, we have the riboflavin, the niacin, the pantothenic acid, the B6, biotin, folate, and the vitamin B12. On the other hand, we have the fat-soluble vitamins, the A, D, E, and K, that is involved in plasma membrane processes, more hydrocarbon-like with fewer functional groups, and occur in the lipid fractions of their sources. Their molecules have double bonds or phenyl rings, so oxidizing agents really attack them and destroyed by prolonged exposures to air or to the organic peroxides that develop in fats and oil turning rancid. That is why if we try to smell these vitamins, usually if it's destroyed already, it turns rancid. Because of the fat soluble vitamins are easily oxidized, they destroy oxidizing agents which are involved in the development of the coronary heart disease, genetic mutations, and cancer. First, we have the vitamin A. Vitamin A is a primary alcohol of molecular formula of C20H30O that occur only in the animal world, where the best sources are cod liver oil and other fish liver oils, animal liver, and dairy products. The provitamin A is found in the plant world in the form of carotenes. So the provitamins have no vitamin activity. However, after ingestion in the diet, the beta carotene is cleaved at the central carbon carbon double bond to here, cleavage here, to give a molecule of vitamin A. That means we cannot obtain directly vitamin A from plants. We obtain only the beta carotene, and once we ingest that beta carotene, it will be cleaved at the central carbon carbon double bond to give two vitamin A's. Okay? Next, functions of vitamin A. It is for vision. In the eye, vitamin A combines with opsin protein to form the visual pigment rhodopsin, which further converts light energy into nerve impulses that are sent to the brain. That is why it aids for the vision. Regulating cell differentiation, a process in which immature cells change to specialized cells with function. Next, maintenance of health of epithelial tissue via epithelial tissue differentiation. Lack of vitamin A causes skin surface to become drier and harder than normal. Next function is reproduction and growth. In men, vitamin A participates in sperm development. In women, normal fetal development during pregnancy requires a vitamin A. Next, we have vitamin D, or the sunshine vitamin, the anti-rachitic vitamin, necessary for a normal calcification of bone tissue because vitamin D aids for the absorption of calcium in the body. It controls correct ratio of calcium and phosphorus for bone mineralization, or what we call it hardening of the bone. We have two forms of active in the body, the vitamin B, D2 and D3. The pigment in the skin, the 7-dehydrocholesterol, is a provitamin D. When irradiated by the sun, becomes converted to vitamin D3. Human exposed to sunlight year-round do not require dietary vitamin D. So that means I would like to correct the notion that we've learned from our elementary days and high school days that when we go outside and have the sunlight, we can obtain vitamin D, and generally we were told that if we want to have vitamin D, I mean, we need to expose ourselves to sunlight because sunlight provides vitamin D. And that is wrong notion. I would like to correct it that we produce vitamin D with the help of ultraviolet radiation, wherein the ultraviolet radiation strikes to our skin, it will it will convert the vitamin D and it will be absorbed by our body. So that means human exposed to sunlight here in a year round do not require dietary vitamin D because our body produces vitamin D with the help of that sunlight, of that ultraviolet radiation that strikes on our skin. Hopefully, we correct the previous notion that we've learned from our elementary and high school days. Next is the vitamin E, the anti-sterility vitamin. And the generic name is the alpha tocopherol, is most active biological active form of vitamin E. The tocopherol, the Greek word which is the promoter of childbirth, and it functions in the body as an antioxidant in that it inhibits the oxidation of the unsaturated fatty acids by oxygen. Primary function is for antioxidant. It protects against oxidation of other compounds. That is why it is good for the skin. That's why Myra E, for example, I do not intend to um, I do not intend to advertise product, but I would like to give you an idea that you are familiar of. So that is why I mentioned Myra E. Myra E is a vitamin E, okay, or the alpha tocopherol, and that is said to be anti-sterility vitamin. One example of this um, vitamin E activity is that they used here in Kalinog, they used the alpha tocopherol supplement to the rag chicken, as to the rag chicken can produce a good sperm for for the for the production of the rag in West Visayas State University, Kalinog campus. That is actually headed by Professor Selma Casiple. And thesis researchers are actually under Ma'am Selma. It was already performed here in Kalinog campus. Another is vitamin K, and it is said to be anti-hemorrhagic vitamin. The vitamin K is synthesized by bacteria that grow in colon and usually active in the formation of protein involved in regulating blood clotting. That is why this is a blood clotting vitamins. Deficiency may occur during the first few days after birth because newborns lack the intestinal bacteria that produce vitamin K and because they have no stored vitamin K in the body. Deficiency may also occur following antibiotic therapy that sterilizes the gut. Okay, again, vitamin K is produced by the bacteria grown in our colon. And that ends the presentation. Hoping that you understand all the concepts that I've been mentioning earlier. And if you have questions, you may be able to contact me through my email or through my Telegram. Okay, so again, thank you very much and happy learning, everyone.